Welcome to Biology 2420, which is Introduction to Microbiology. I am Dr. Paula Nelson. We are using the OpenStax microbiology book that is available through the openstax.com, which is provided through Rice University. So I will be providing these PowerPoints for each chapter and doing kind of this voiceover for the videos, just as if you were in my face-to-face class. So obviously we're starting with chapter 1. It's called The Invisible World. And basically it's like, what is microbiology? A lot of people come in with different preconceived ideas. Mostly students come in thinking, oh, this is going to be such a hard class, and I've been dreading it. Trust me, it can be a lot of fun. We are looking at a lot of different things. Microbiology encompasses studying any organism that is too small to be seen with the naked eye. So there are a lot of things that can fall under this category. Things such as bacteria, which is one thing that most people think of. Archaeas, a lot of people aren't really sure what those are. By the end of this course, you will. They're small prokaryotic cells, similar to bacteria. They're actually, we'll get into talking about characteristics of them at a later chapter, but just so you know, they're often considered to be the first living organisms that were here on Earth. And then we also look at fungi, we'll look at viruses, we'll continue to look at algae, and then also protozoans and helminths. And a lot of people go, what is a helminth? That is a broad category, basically worms. It includes nematodes, it includes flatworms, it includes flukes and tapeworms. But all of those you have to look under the microscope in order to see them, say in a patient's specimen sample. And so that's why it's included under microbiology. So most people, when they're first introduced to microbiology, are like, ooh, germs. Don't call them germs to microbiologists, please, because they're not all bad. Only 5% of the known microorganisms are pathogenic or disease-causing for humans. That means the other 95% are good, or, no offense, they don't really care about us. They're not going to bother us either way. So I often say, if you compare the microorganisms, which oftentimes we just call microbes, in man, are they friend or foe? Which are they? It depends on how you look at it, and it depends on which ones you're looking at. A lot of people with microorganisms think of them, as I just said, as pathogens, as disease-causing. Oh, they're out to kill me. That's not true, as I just mentioned. And there's different areas of microbiology. There's a huge number of subspecialties. It's not all just medical. They play a role in agriculture. They play a role in the environment, in food, in industrial applications, as well as in the medical field. So if we look at it just briefly as just kind of an informational thing as to what are you getting yourself into. Just briefly, now in this class we will focus mostly on medical aspects, but just to let you know, in terms of agricultural microbiology, we look at microorganisms in terms as pest management. Some of them, like some bacteria, are actually used to control other pests so that you don't have to spray chemicals. That's the good side, and there's a good side and a bad side for everything. On the bad side, there are a lot of microorganisms that are plant pathogens, and so that's of concern to the farmer who's trying to grow crops, and his livelihood is dependent on the production of whatever type of, say, grains or produce, etc. And there's concerns about pre-harvest versus post-harvest. Pre-harvest, will there be maybe fungi that attack plants and destroy it and cause rotting before you ever get the production of your, say, strawberries or your tomatoes or your rice or wheat or whatever? Now, if you manage to grow the plant and harvest your product, now you got the flip side of what we call post-harvest pathogens. Some that are now, well, I just was hanging around waiting for you to get those nice juicy strawberries because I don't care about the plant, but I want to infect the strawberries. So, like I say, everything has a good side and the bad side. Some microorganisms are used for that integrated pest management, trying to reduce our dependency on chemicals that are used to manage plants. But then on the flip side, we do also have microorganisms that basically are pathogenic to the plants. In terms of environmental microbiology, what we're talking about here are how are the microorganisms involved with nutrient cycling. We'll see how recycling nitrogen and carbon and sulfur and all of those various nutrients. Some microorganisms are used in bioremediation. I personally did a lot of research in this area where you use a microorganism, specifically I was using soil fungi, to use to break down hazardous chemicals, in my case that were in the soil. Some organisms are in a symbiotic relationship. Lichens that you often see, we'll talk about those, and mycorrhizal lichens are a symbiotic relationship between algae and fungi, and then mycorrhiza are between fungi and plants. So here's just an example of how we have used, in environmental microbiology, we have used microorganisms in different applications. This is from the Deepwater Horizon oil spill that occurred in the Gulf of Mexico in 2010. There was a leak, and there was a lot of oil that was spilled into the Gulf. It did make it towards the shorelines. And so this is just showing someone who is involved with helping to try to rescue the animals that are coated with the petroleum products. As far as in the water, they did use a bacteria that they were able to basically put it in the water, and it breaks down the petroleum products. So it was very successful. This had been used previously in the Alaska oil spill, then the Exxon Valdez accident that occurred years earlier. It was fairly quick in terms of the cleanup that occurred. Warmer waters in the Gulf of Mexico speeded up the cleanup process. In terms of food microbiology, a lot of microorganisms are used in the production of food. We use yeast, which is a single-cell fungus, to help make bread, to help make cheese. We definitely have learned how to use the microorganisms, which organism to use with, say, which fruit or grain product to produce alcoholic beverages through fermentation processes. Something that a lot of people are not aware of is that we also use microorganisms to make a lot of the flavorings that we add to food items. So this is just showing, over here is the yeast, which is a single cell fungus, and then this is what it looks like under an electron microscope. And then here you have, if any of you do baking, you're familiar with, you take the yeast specifically for bread, usually it's Saccharomyces cerevisiae, and you put it, typically to activate it, you add warm water or warm milk and some sugar that activates it, and then you use it to make your bread. From an industrial standpoint, sometimes this is involved with manufacturing different compounds such as enzymes, various chemicals, various products that are used in a whole wide range in the field. Manufacturing enzymes, things like, if you're thinking what enzymes are used, probably a lot of you have heard or seen commercials where they talk about detergents and, oh, this brand of detergent has an enzyme that will break down the grass stains. Where do they get the enzyme from? They get it from microorganisms. Some of the chemicals production and things that you routinely buy, like isopropyl alcohol. Isopropyl alcohol is rubbing alcohol. We use microorganisms to make that. In terms of other products like genetic products, human insulin, we use microorganisms now to produce human insulin, human growth hormone. We'll have a chapter where we'll talk about the applications of some of this, what's known as recombinant DNA techniques. And so you'll learn how we do that. Also involved with the industrial microbiology subfield, if you will, has to do with wastewater treatment and drinking water treatment. You'll find out that a lot of diseases are caused by contaminated water. So we need to be able to, any wastewater, we need to put that through a plant and clean it so that we are not adding contaminated water to our normal regular water waterways like rivers, creeks, lakes, the ocean, etc. And then also we want to make sure that our drinking water supply is not contaminated with microorganisms. So there are processes of which trying to make sure that that stays clean. And then the medical, as I said, is the primary focus for this particular class, where you'll be studying about the pathogens, which microorganism causes which disease. We'll also learn about some of the beneficial microorganisms. Whether you want to think about it or not, we're all covered with microorganisms. They are supposed to be on our skin. They are supposed to be in our mouth or in our nose. This is helping to protect us. If the space is already occupied with a beneficial microorganism, then there's nowhere for a pathogenic one to set up shop. And so even though you might not want to think about it, yes, we are covered with the beneficial microorganisms. That's the way it's supposed to be. We'll also, from a medical standpoint, talk about those microorganisms that naturally produce antibiotics, which are chemicals that will inhibit the growth of other microorganisms, which of course we have also taken this and exploited it, if you will, so that we can use that to control the growth of pathogens, and we use it for treatment purposes. And so with the medical aspect, this is an example of someone who's having a lumbar puncture. They are removing some of the cerebral spinal fluid, which is fluid that bathes both the brain and the spinal cord. They are removing some of this to test for infection. This should be clear. It should be sterile. There should be no microorganisms, no bacteria, no viruses, nothing growing in it. And so this is a test. If it's cloudy, that's a problem. It should be clear. If it's cloudy, that's kind of indicative that there is a bacterial infection, and you better start some treatment right away. So once again, when you look at microorganisms, are they a friend or a foe? What is the economic impact of microorganisms? You could discuss from a positive side the amount of benefits from microorganisms and the attributes that they add towards our economy in terms of food and whether it's chemical products. You can also look at it, how much money is put into or how's the economy affected by the disadvantages, by illness, by diseases, the amount of money that is put into trying to control or inhibit the growth of microorganisms. But what I would challenge you is that you cannot go a single day without having some interaction with microorganisms. So a lot of this chapter looks at from more of a historical aspect of microbiology, kind of what did our predecessors, what did our ancestors know? They knew that microorganisms were useful for making fermented foods. They knew that they were responsible for diseases and somehow with the spread of diseases. The Romans, back in Roman time, they even built sanitation infrastructures. Kind of had a knowledge that our wastewater, we want to get it away from where we're growing food and where we're drawing our drinking water from. They also knew that what you consider very basic immunity factors in terms, they know if you got sick with something and you survived, you usually didn't get sick with that same disease again. They didn't necessarily know why these things happened, but they picked up on some of these. They were very observant. The father of microbiology is considered to be Antoine von Leeuwenhoek. As you can see, he lived in the 1600s, early 1700s. He was a Dutchman by trade. He basically worked with fabrics and clothing, and he was not a scientist by training whatsoever. He was the first to view microorganisms. He referred to them in his notes as little animalcules and little wee beasties. He took very meticulous notes, which we still have today, some of his writings and drawings. This is an example of the microscope that he made, which basically is like a early magnifying glass, but he was able to put a sample and observe it. And like I said, he noticed these very small critters, if you will, that were in the various samples. You know, he was an interesting person from the standpoint, he, like I said, was not trained in science, and he would write up papers, send his papers, his manuscripts, to the Royal Academy in London. Consider the time that he was living, late 1600s, early 1700s. There was a lot of nationalistic pride. There was, and still is, egos, if you will, some arrogance associated with some individuals. So when the members of the Royal Academy of Science received his manuscripts, they looked at it and they thought, this man is crazy. What is he talking about, these little wee beasties in the water and samples? Is this man lost his mind? And who does he think he is? He's not even trained at any of the big name universities like Oxford or Cambridge, and you know, he's not one of us. And so they'd reject his manuscript. And then he'd be observing other things and send in another manuscript. And they're like, finally it was kind of like, this crazy Dutchman, here's another paper. And so unfortunately, like we often see, is that he was not recognized in his lifetime for the many accomplishments that he made. He also became very bitter, and he did not teach anyone else how to make those little microscopes. You couldn't just go buy them at Walmart or off eBay. You had to make them yourself. No one to this day has been able to grind the lenses that are used in there. No one's been able to grind lenses to the accuracy and precision by hand that he was. And so it kind of makes you wonder if his contributions were acknowledged during his lifetime, where might we be with our knowledge in the science? How much did that delay the progression of the field of microbiology? Because people refuse to accept and acknowledge his contributions, and that's a shame. There are some of his papers that have been translated. I have a book that has some of them. One of them was kind of interesting, a paper where he was just a very curious man, and he took samples of scrapings from his teeth to see what was growing there. And then he was smart enough to say, well, let me compare this with somebody else. So that must be a weird thing to go to a friend and say, you know, hey, can I get some scrapings off your teeth because I want to look at it under the microscope? But that's what he did. And it's kind of interesting reading it because the friend he went to, he quoted it as, was known to imbibe in alcohol. Well, he was wondering if that might make a difference in what he saw, and it did. Another friend he went to, he found differences again, and in the description of that person, said that the person had not brushed their teeth for, I believe it was 3 weeks. Which I must say, the microbiologist in me is saying, Oh, I bet you found some stuff on there. And of course, the mother side of me is going, ew. So, yeah, I bet you found stuff on there. But he's considered the father of microbiology. Now, later we had these two individuals, Louis Pasteur and Robert Koch. They are certainly during the time period that's known as the golden age of microbiology. There were so many discoveries made during this time period that really, really advanced the field. And Louis Pasteur and Robert Koch were during the same time period and kind of, in many ways, competing against each other. One would discover something, and then the next, while the other guy would discover something. Once again, in this time period, there's a lot of nationalistic pride. So Louis Pasteur was French, Robert Koch was German, and they were both highly supported by their individual governments. So it was like Louis Pasteur would discover something, and then the German government would say, here, Robert Koch, here's some money. We need you to go look and, discover something. We got to beat Louis Pasteur. And so then Robert Koch would discover something. And then the French would give Louis Pasteur money and say, come on, you can't let the German beat us. We've got to, from a science standpoint, or I guess I should say a scientist standpoint, that's kind of an unusual situation to be having the government throwing money at you to support your research, which would be nice, but it definitely helped with this explosion of new discoveries, and overall certainly helped things, the overall quality of life, finding out a lot during this time period what organism caused what disease, how to perfect the fermentation process for, say, production of wines to keep it from getting contaminated. There were so many different things that were done during this golden age. Sometimes they talk about a systemic approach to studying things. Linnaeus developed this classification system. You may not have heard of them. You may have, depends on if you've had other biology courses. If you've heard of them, you may be familiar with the classification system that he came up with of looking at all living organisms and giving it a genus and a species name. Species can, oftentimes if you're talking about like animals, they can meet. The genus is, you're related, and you start off with domains and kingdoms and then go down to genus and species. So what we call binomial nomenclature is genus and species. Just so you know, when you write that out, it is italicized. Genus is always capitalized. Species is not. I will not be overly picky about that, but just to let you know, some people are. I had a professor when I was in school that you could write the name, spell it correctly, and you're going to see some of these names are rather long, but if you did not italicize it, or if you're handwriting it, you underline it. If you did not do that, it was wrong. And you're like, man, I figured out where the I's and O's and C's go, and now you took off because I forgot to underline it. So there is this whole field of taxonomy, which is classification of living organisms. I will say this, the field of taxonomy has changed quite a bit over the past 30 years as our improvement of DNA techniques has also greatly improved. So you may see, oftentimes if you're reading, things have been shifted around a little bit with our classification systems. So you might see where here's organism A, previously known as BB, also known as CC, because they've been switching things around a bit. A phylogenetic tree shows evolutionary relationships between organisms. Oftentimes that's important to see kind of who's related to whom. The classification scheme, the taxonomy, as I said, has changed over the years. So the current one is that there are 3 domains. All living organisms are placed in one of these 3 domains. The archaea in the bacteria are prokaryotic cells. These are the simplest cells. We'll talk about those later, what that means to be a prokaryotic cell. But just think of them as smaller, less complex, earlier in terms of evolutionary standpoint. The archaeas were here first, then the bacteria. The eukarya are what we call eukaryotic cells. This is then broken down into kingdoms. You have the Animalia, the fungi, plantae, and protista. So these are going to be larger organisms, have larger cells. They're a little bit more complex, and essentially they're the new guys on the block. But every living organism will be placed into one of these three domains. And as you saw on a previous slide, in microbiology, we focus mostly in this class on the bacteria, but we also will look some at the archaea. And under eukarya, we will be looking at fungi and protistas. So this is often how the phylogenetic tree is organized or shown, where you have the three different kingdoms. Something that's on the same branch is more closely related than something that's over on a far different branch. So to classify bacteria, there are two different manuals that typically are used. These are the classic standby Bergey's Manual of Determinative Bacteriology and Bergey's Manual of Systemic Bacteriology. They will list every single bacteria that's ever been discovered, and it places it in its categories. I do not own my own copies of these because they're actually, I believe it's 4 or 5 different volumes, which are about $400 a piece. So I don't own my own personal copy of this. If I have to look something up, I know where to go to look it up for free instead of buying the whole set. To classify bacteria, we often will start by doing some staining, and then we do a lot of biochemical tests. We may also do serological tests, which is blood testing, how it reacts with different types of reactions in there, and also DNA and RNA analysis with it. But the first thing will be to do some staining, look at it under the microscope to get an idea of the size, the arrangement of it, how it reacts with certain stains. But we do not, as microbiologists, spend all of our life looking through a microscope. We actually probably spend more of our time doing biochemical testing. So when we look at microorganisms, we look at prokaryotes versus eukaryotes. There are other things that we look at and how they're different. We look at sizes, the shape, characteristics. Where does it grow? What habitat is it in? What type of chemical reactions or metabolic activities can it carry out? What are some of the growth requirements? Does it need oxygen? Some yes and some no. What type of carbon source is it able to use? Can it break down glucose? Can it break down lactose? pH. Some organisms prefer to grow at a more neutral pH of around, say, pH 7. Others prefer more acidic, maybe a pH of 5. So we look at all these. You take note of it. One thing about being any scientist, not just a microbiologist, is being very observant in noticing all of these things. I'm always telling people, especially in the lab, when you're making these observations, I like to do puzzles. And so I often describe it as, it's like somebody has given you a box with 1,000 pieces of a puzzle in it, but you don't know what the picture's supposed to be. So why don't you start with what I call the straight edges, the ones that are going to form the margins or the edges, the frame. Put those together first. And what you're trying to do is, you know you have a puzzle. Well, okay, you know you have a microorganism. What is it? Well, every time you do a test or you make an observation, it's like putting another piece of the puzzle. And you're starting to see slowly, what's the overall picture of this puzzle? The results of all these various tests you're going to do and what it looks like. Each one of those things is a puzzle piece to give you an idea of what organism is it that you're dealing with. The size. In microbiology, we're on a whole different framework in terms of size of things. Obviously, we cannot see bacteria or viruses or fungi with the naked eye. And so this gives you a little bit of an idea. Over here on this side, it shows the frog egg. It's about 1 millimeter. Most people think, oh, that's pretty small. Yeah. To a microbiologist, that's big. Okay. This is the largest end on this side. As we move down here, things are getting smaller and smaller. So now you're seeing an animal cell or a plant cell. Now you're seeing a red blood cell. Way down here is a bacteria. You can see a bacteria with a light microscope. Now it's going to look very, very small. When you look, and you'll see pictures a little bit later on, when you look, it's going to look like little tiny dots, or just little teeny tiny, we call them rods, little straight lines. You can see that with a light microscope. In order to, the best that you can see it, the highest total magnification you can have with a light microscope is 1,000 times. So you'll be able to see bacteria, you'll be able to see fungi with that. Can you see viruses? No. You would have to use an electron microscope in order to see any of your viruses. And certainly if you move down the chemical level of seeing atoms, there's no way you can see that even with an electron microscope. We have to go to something even smaller. So to a microbiologist, like a plant cell is huge. I used to laugh and tell my kids when they were really little that if they were wanting information about stuff, they had questions. Well, I said, Mommy's used to dealing with stuff you can't see. If you can see it, go ask Dad. If you can't see it, then come ask Mom. Unless it was human biology, because I teach also A&P. So if it's anything with the human body, then I was like, yeah, you can ask me. But we're just on a total different size frame. Some people, that's a little bit of a stumbling block because they're used to, especially if you're a very tactile learner, you're used to touching stuff. Let me see, how does it feel? And you can't do that in microbiology. And my easiest advice to you is just accept it. Just try not to get bogged down on that. Just accept that it's small that we're looking at. I always threaten to sing, it's a whole new world, but you don't want to hear me sing, so I'll save you that punishment. But there is this whole fascinating world that we can't see, and that's okay. And can you touch it? No, not like you're used to, and that's okay. Just accept it. It's okay. So we look at the archaeas. These are prokaryotes. What does that mean? It means that in these cells, a prokaryotic cell does not have a nucleus. That's the main thing. It has DNA. Don't get confused by that. It does have DNA, but the DNA is not found in a nucleus. It is single cell. Now it does, archaeas do have a cell wall, but they do not have the chemical peptidoglycan, and that's going to become important later. I mentioned, I believe a little bit earlier, is that from an evolutionary standpoint, we feel the archaeas were the first organisms on Earth. They tend to be found in very harsh environments, environments where you would think, oh, nothing can grow in that. Well, yes, they can. They're there. They can grow in environments with very low oxygen. They can grow in very acidic environments, environments with a low pH. They can grow in environments with a lot of sulfur. So something where you're like, oh, that is a nasty environment. Well, there might be archaeas there. From a medical standpoint, none of the archaeas are human pathogens. So that part is good. Because they are able to grow in a very harsh environment, we are starting to kind of revisit them, relook at them as organisms that maybe what we're finding are beneficial in terms of breaking down some of our pollutants. Like I say, they grow in these harsh environments. I don't know if any of you have ever been to Yellowstone. This is a picture of some of the hot springs in Yellowstone. It's kind of a famous picture that they have them. The different colors around here. This is due to some of the archaeas that are growing. They can grow in that really hot temperature, which would kill most anything else. But if you've ever wondered what is the coloration here due to, it's due to the archaea that are growing there. So this beautiful picture is brought to you by microorganisms. The other group of prokaryotes are in the domain bacteria. Once again, these are also single-celled or unicellular. The cell wall for bacteria does contain peptidoglycan. This is one way you can tell the difference between the archaeas and the bacteria. The bacteria are found in all environments. They have been found in the water, deep surface, the bottom of the ocean. They're found on land. They're found in the air at 30,000 feet. So they're everywhere. When you look at them, there's a huge diversity associated with them, different sizes, different shapes, different abilities to carry out chemical reactions. So their metabolic capabilities is extremely diverse. As I mentioned earlier, only about 5% of them are known as human pathogens. So the other 95%, don't worry about it. We start off by looking at the morphology, the shape. Morphology means shape. So that's also going to be an important factor to look at for classification of them. So these are showing just some of the common shapes, and get used to it. We're going to come up with different vocabulary for you because, you know, it's just too easy to say circle or rod. So in microbiology, instead of saying spherical or circle, we say coccus. Instead of saying rod shaped like this, we say bacillus. Now I will caution you. On this particular diagram, they have capitalized all of the terms. Usually one thing to be careful of, bacillus with a lowercase b refers to rod-shaped. Bacillus with a capital B actually refers to a genus name of certain genera of bacteria, so kind of watch that. Sometimes we're not real creative with coming up with new names, apparently. Vibrio is kind of a comma-shaped. Coccibacillus, this is where, well, it's not a perfect circle, but it's not really a rod yet. It's kind of oval-shaped because it's a cross between the cocci and the bacillus. So what do we do? Because we don't know which way to classify it, we give it a new name by just combining these two names together and saying, oh, that's a coccibacillus. Spirillum is kind of curved like this, and then spirochetes. Spirochetes, to me, they look kind of like a spring or a slinky almost. They're very distinctive in how they look. So that's an example of what some of the shapes look like. So then you now have the eukaryotes. Eukaryotic cells in the domain eukarya, they appeared more recently than the prokaryotes did. They can be either unicellular or multicellular, depends on just which one you're talking about. They have both. Usually they're larger, the cells are larger than prokaryotic cells, The distinguishing thing is that they do have a nucleus. The DNA is inside the nucleus, which is inside the cell. It adds an extra layer of protection for the DNA. Nucleus in this case is just, usually it's around the center of the cell. It also may have membrane-bound organelles, including things like mitochondria, endoplasmic reticulum, Golgi apparatus. Those are present in eukaryotic cells, but are not present in prokaryotic cells. In the eukaryotes, when we look at some of the specific ones, we look at the kingdom of Protista. The Protistas, they're a very diverse group because they're kind of the catch-all kingdom. If you're not a plant, you're not an animal, and you're not a fungus, but you are a eukaryotic cell, meaning you do have a nucleus, where do we put you? Well, we throw you into the Protista kingdom. They're usually found in a moist environment. It includes things like algae. It includes things like a lot of your protozoans. Protozoans usually are motile. Algae are usually photosynthetic, so they're distinctive from that. A type of algae are your diatoms. They're usually wide, diverse group of shapes here. They look really kind of cool. All of these are different types of diatoms. As you can see, they're different sizes. There's small ones here and these large circular. So they're really kind of cool looking. This particular sample is taken from Antarctica in the Bermuda Sound. Excuse me. They are, as I said, classified with the algae. Here's another type of a Protista. Specifically, it's a protozoan. This one is Giardia. Not sure if any of you have heard of it. If you drink water that has Giardia in it, you're going to regret it later, because you will end up with what a professor of mine used to say, severe intestinal distress. You're going to have severe diarrhea, and you're going to wish you had not drunk that water. This is oftentimes the cause with contaminated water of why they have to put a boil water order in places, or certainly if you are someone who goes camping or backpacking, why you don't want to just drink unfiltered water from a stream. You want to be able to boil it or filter it, because if this guy's in it, you are going to be sick. So that's what Giardia looks like. Fungi. One of the other kingdoms of eukaryotes. Fungi are not photosynthetic. Okay, years ago they used to always group fungi. When you studied botany, when you studied plants, you always studied fungi, which never made sense to me because they don't carry out photosynthesis. My husband used to be a botanist, and he was like, oh yeah, we always study fungi. I'm like, no, you study your plants, but the fungi are mine. These are microbiology, organisms. They don't carry out photosynthesis, so that's the first major thing of there's no chlorophyll in them. They're not a plant. They are very, very important in nutrient cycling because they're decomposers. So they will break down a lot of decaying material. They do have a cell wall. Their cell wall is composed mostly of chitin. So already if you've picked up that cells that have a cell wall, there's different chemicals in it, and you can use that to help to identify what is this that I have. Now, these are eukaryotes, so they will have a nucleus in them. If they are single cell, we refer to them as a yeast. If they're multicellular, it's mold. So both yeast and mold are types of fungi. Fungi are the #1 cause of diseases in plants. So gardeners, farmers do not like them usually. So this is showing a picture of kind of albicans, which is yeast, It can cause several different types of infections in humans, but you can see it has these little yeast, typically are these small little circles. Here are some individual ones right here, and then they tend to bud when they reproduce. One just kind of buds off the other, and so you see these clumps of several yeast cells clumped together. And as I said, because they often do cause diseases in plants, that is a concern in the farming industry. When you have a large group of fungi together, then you can see them with the naked eye. You may see a clump of the spores. If you've ever had strawberries and you get that white fuzzy, because you kind of forgot the strawberries in the fridge, and then you get the white little fuzzies on it, that's the spores from fungi. In this discoloration here, that's also fungal infection on there. What you're seeing are literally thousands and thousands of spores or cells in there, not the individual ones. Another group of the eukaryotes that we study are known as the helminths. This may be one term you're not familiar with. Helminths, it's a large category. Basically what they are parasitic worms that are multicellular. So they're often associated with contaminated food or water. And the reason we study them is that the eggs and the larvae are microscopic. Sometimes the worms themselves are not, but they go through different stages in their life cycle in the eggs and the larvae, which is often how they are ingested by humans. We're talking about things like nematodes, tapeworms. Most of you, if you saw a big long tapeworm, you're not going to eat whatever it's in, but you may not see the egg or the larvae in it. So that's why we study them. So over here is under a picture is a tapeworm, very long, and then over here is another type, it's known as a guinea worm. Usually for those, to remove them, you don't want them breaking off internally, and so oftentimes what they'll do is they'll take a stick or a rod, and once they get the end, they just slowly roll that to carefully remove it from the individual. Viruses, we also study viruses while we're in here. Viruses are acellular. Viruses pretty much composed of, it has nucleic acid, meaning it has nucleic acid. It's either DNA or RNA. And I'll tell you right now, viruses do not have both. They have one or the other, but never both. They will, excuse me, they will have that either DNA or RNA, and it's going to be surrounded by a protein capsule that helps to protect it. That's it for a virus. It's not very much to it. The other thing when we study viruses, just kind of give you a preview, the DNA is either double-stranded or single-stranded. And the RNA is either double-stranded or single-stranded. So we'll use that for classification purposes. Viruses are interesting in that they're very host-specific. So there's lots of viruses, but it doesn't mean that all the viruses are going to infect humans. What happens is they're host specific because the virus needs to attach typically to certain proteins on the outer surface of the host cell. So the proteins that are on the surface of, say, human cells are different than, say, on a plant. So that's why usually a plant virus is not going to infect a human. And even within humans, our cells are different in different locations. So that's why, like the influenza virus is very specific even within the host of humans as to where it is going to attach. An influenza or the flu virus prefers the respiratory system. That's where the receptors are that it can bind to. That's why influenza virus on your skin doesn't do anything, because it can't attach to it. All of you, whether you want to or not, are familiar with the coronavirus now from COVID. that is also going to prefer the respiratory system. Will it do anything on your skin? No. So it's very host specific, and not only is it host specific, it's specific where on the host it can attach to. And why is it so important? Is because a virus cannot reproduce by itself. It has to reproduce with the host. It has to infect the host cell, and then it takes over that host cell so that it can reproduce itself. Because of this, just so you are aware, and it will depend on which book you read and who you talk to, there are still two different schools of thought within scientists, whether they're biologists or whether they're microbiologists. There's two schools of thought. Some people feel as though viruses are living, and some feel they are not living. And the whole controversy of this comes from where the reproduction occurs. So some people say viruses are living because they can reproduce, and that is one of the characteristics of a living thing, is that it has to be able to reproduce. But other people say, it can't reproduce by itself. It has to have that host cell to reproduce. So therefore it's not living. So you can look at that and figure out which way you feel. There's not a right or a wrong. I would say as long as you justify your answer, there is not a right or a wrong. Just be aware that there are these two different schools of thought. Relative to COVID, just kind of FYI, that is why during the huge pandemic back in 2020, when they were trying to do social distancing and quarantine, the idea was, and with the vaccines, part of it was what we call herd immunity. If we can prevent that virus from infecting new people, then slowly your numbers go down. If it doesn't have a host, it can't reproduce. So over here under A, this is the coronavirus. Coronavirus is a family name for viruses, so it's not just what you're familiar with COVID. It can cause the whole family. There's several different types of the virus, known as SARS, MERS, etc. But that's what it looks like with the little, you can barely see the little spikes sitting off of it. Always when you look at a picture, look at here in the legend so you can see size-wise. This is 50 nanometers, so this is really, really, really tiny. Over here is the Ebola virus, the causative agent of Ebola. So within microbiology, we have a lot of different specialties. Someone may want to just study bacteria, so that'd be bacteriology. Mycology is study of fungi. Virology is a study of viruses. Parasitology is studying parasites, so it could be some of your protozoans in there. Immunology, specifically looking at the human response to infection. You may look at agricultural, you may look at genetics. Physiology, how are things working? Protozoology, which is looking specifically at protozoans. Ecology and environmental, what is the effect on the environment? How are things being broken down? Epidemiology is studying basically disease outbreaks and tracing. Every state has a state epidemiologist who helps to monitor any outbreaks of diseases. Who was the first person? Can we trace back to how they initially got sick? Let's say if it was because of food contamination, then we need to fix that so nobody else gets fixed. There is a professional society known as ASM, American Society for Microbiology. And I have been a member of that since 1983, so a long time. I started out as a student member. When you are a member, one of the things you get to pick are two basically subspecialties, two areas of interest for you. There are so many specific specialties. They label them alphabetically. So you pick, oh, I want to be in part A or C, or they're in double letters now because there's so many different subspecialties within microbiology. There is no way that one person is going to be an expert in all fields of the microbiology areas. I have kind of a unique background. That part of my background has been very strong with medical microbiology. I've done a little bit of research in some of that, but then I also have a background in environmental microbiology. So most people are one or the other, but I kind of covered it all. So my specialty area really is microbial physiology. I love looking at, I know it probably sounds boring to a lot of people, and so I'll just confirm that yes, I'm weird, that I love looking at how are they able to break things down. It just fascinates me that things that are so small like bacteria, but the regulation and the control that goes on at the cellular level to be able to, whether it's breaking down glucose, whether it's breaking down lactose so that it can get energy, and how is it able to respond to environmental changes and adapt to it. I just, to me, find that fascinating. So this first chapter is giving this kind of brief introduction and brief history of what we're going to be looking at. Basically, we're looking at really teeny tiny things. You're not going to be able to touch it. Just accept it. It's okay. We'll have fun while we're looking at this. We're going to be looking at things, excuse me, such as focusing on this first bit, just some general background as far as the cell and what are the components of the cell, more detail into the eukaryotes versus prokaryotes. We're going to be looking at, with a microscope, like how do we see things, how do we stain things so we can actually see it. We're going to move into looking at, it will involve some chemistry, don't panic. Chemistry is not a prereq, but we will look at some chemistry as it relates what you need to know for the microbiology that makes sense. We're going to look at how do microorganisms, say, break down glucose to get energy. How do they grow? How do we control their growth in the environment? How do we control their growth within us? So like using antibiotics. We're going to look at genetics. How does the DNA replicate? How do we use a lot of modern techniques and applications? When we look at, say, can we use E. coli to grow and produce human insulin? How do we do that? We'll get into that. We will look into a section on epidemiology, how things are transferred, how do we study, what's the difference between a pandemic, epidemic, endemic, sporadic outbreaks. Some of that, unfortunately, you probably know because of COVID. We'll also look into the immune system. How does the body respond? We look at two different aspects. Microorganism trying to invade the body versus how does your body respond to that if it's if it's any organism. How your body responds can help prevent disease. So is something good or bad? Well, it depends on your point of view, because if you're looking at it from the viewpoint of a microorganism, if it's able to penetrate through their skin, from a microorganism standpoint, ooh, that's good. From our standpoint, not so good. Once it's in the body, okay, now we got to do something else to try to prevent infection. If you're able to attack and destroy it, hey, that's good for us, not so good for the microorganism. So you're looking at it from two different angles. We will then continue on and look at some of the diseases of the immune system. How do we test different things with the immune system? And then this semester is divided into 5 units. The last 2 units are where we will actually look at specific diseases. This book is divided a little bit differently than when I'm used to teaching it, but that's fine. It's divided by systems. So we'll look at the systems, like we'll look at first skin and eye infections, and then we'll look at the respiratory system, and after that the urogenital system, and then digestive system. So for each of those systems, in the later chapters, we look at the system, and within the system, we break it down by, here are some of the bacterial infections, here are some of the fungal infections, here are the viral infections, here are the protozoan infections. Some of the chapters, I can tell you right now, are very detailed. Don't worry, we're going to be focusing, this is an intro class, and I'm always trying to balance that. We'll be looking at more of, this organism causes this disease. This organism causes this disease, like in, say, a matching type section. So we will cover all 26 of the chapters, but this is the first one down, and hopefully we'll have fun. You'll learn a lot from this class, and you'll find out at the end, it hopefully is not quite as scary as what maybe you thought it was going to be.